Hi everyone. So Dr. McNew was born and raised in New Mexico, which is maybe the southwestern area of the U.S., an important area of interest. She earned her bachelor's at Pomona College in California, and during her undergraduate, she studied abroad in Costa Rica, where she was able to learn Spanish and get field research. She then worked as a fellow under the Fulbright Commission of Ecuador, and then worked as a postgrad researcher at the University of New Mexico. At the University of Utah, she worked as an NSF graduate research fellow and earned her PhD there as well. She then moved on to work as a postdoctoral fellow at Cornell University, and then came here to the U of A to work as an assistant professor in disease ecology.
We have to share microphones because there's only one. So thank you for the invitation. Thanks, Marisa, for the introduction. It's been great getting to know you this summer. And I'm really happy to be here and to see all of you who are having this really neat experience of working as a summer researcher. I feel like it's so different, the feeling of being on campus in the summer and seeing people in labs and getting to focus on that. And it's so different from the experience that we have during the semester when it's all about classes. And so I'm excited to talk to you today about some of the research that we do in my lab, and I hope that you guys are having great summers and getting to know research in the various places that you're working as well. I will start things off by turning down the lights. If I can. Ooh, okay, that's better. I will put this information up again at the end, but if any of you like what you hear today, please take my class. It's in the fall. It's on disease ecology. Anybody's welcome, and I'm happy to chat with you afterwards if you're curious about it. So to get us started today, I wanted to show you a video that my grad student Henry found a few months ago that really captivated us in the lab. And I love this video because it encapsulates so much about what I love in biology, what I find cool and fascinating and gross, and what motivates me as a researcher. So what you see here is a video taken from a game cam in Australia. And there are some wallabies coming to drink at this water source, and then you see these crows coming in, and they have this interesting interaction where if you miss that quickly, it'll happen again. The crow is pulling ticks off of these wallabies. And this video continues for quite a while. These crows are really getting after these ticks, and they're quite large. You can see them on these mammals and these ectoparasites, these big, juicy blobs that are really covering the wallabies. And so this keeps going for a while. So I think this is a cool and gross interaction on a number of levels, but some of the things that I'll highlight are, again, these features in biology that kind of motivate me. So first of all, as a student, I was really excited about animal behavior. So the ways in which animals interact with their environments, behaviors that they change in response to different pressures. And then we also see another one of my loves, which is species interactions. I, again, love seeing how species interact in the environment, whether that be in maybe a helpful way, like a commensal interaction between the bird and the wallaby, whether it be a parasitic interaction between these ticks and the wallabies, and understanding how these interactions change over time is something that is really exciting to me. And all of the things that we see in this video are taking place in the context of anthropogenic disturbance. And that's something that really shapes our world in almost every place these days. And so the modifications of the environments that we make, the habitat destruction that we do, species interactions, all of that shape, or species introductions, all of those things that humans do shape the natural environment. And part of what I'm interested in as a biologist is studying the response of organisms to these features. So Marisa also did a really good job of introducing me, but in this forum I was asked to give a little bit of background. So I'm going to walk you through some fun facts about me through pictures. I am from New Mexico, so I'm a native of the Southwest, liked animals growing up. I went to college in Southern California, and in college, some of the experiences that really kind of shaped my trajectory included study abroad, was a really amazing experience. Even though I was a biology major, I knew that I wanted to have the experience of living in a different country, of interacting with people in a different language, and those of you who might be international students know how just profoundly that changes you and all the challenges that come with having to interact with people and learn in a different environment. But it was also really valuable to me. It really inspired in me a love of Latin America, and at the same time in college, I was gaining experience as a biologist. I started working with birds in college, and that gave me the opportunity to continue doing research down the line. So after college, I went back to Ecuador as a Fulbright fellow, and during that time I was exploring avian research, field research, gaining experience, and also trying to figure out if this is something I wanted to do long term. Eventually that led me to more opportunities working in Latin America with birds, and eventually doing my PhD in the Galapagos, working with birds still. And as my research career evolved, even though I started working with experience in birds and a background and interest in animal behavior, through some kind of up close and personal experiences with parasites, I realized that what I was really excited about was these interactions between hosts and parasites, and kind of the gross, creepy crawlies that affect us, that affect birds, that affect all life on Earth. And at the core of these host-parasite interactions that I really found my interest in science. So I did my PhD, I later did a postdoc, and since 2023 I've been here at the U of A, where my lab studies disease ecology, which is the study of host-parasite interactions. And so if you look at this kind of timeline, something that I wanted to point out in this forum, where some of you might be thinking about, what's next? Is it grad school? Is it something else? I just wanted to say that it can feel like a long time. When I look back, it really did feel like a long and winding road between college and what felt like my first real job. And there were a lot of different kind of points along the way where I was thinking, what's coming next? And I can tell you in total honesty that I did not sit in college and think I want to be a professor. Like I really had no idea. But I knew that I liked science, and over time I knew I liked doing research. And so I kind of stepped through these different opportunities, and each of them gave me the chance to think about the next step. But if you're sitting in your chair and you're not totally sure what your career looks like, that is totally fine. In fact, along the way, there were many moments where I thought, maybe there's something else I might be doing. And I, for instance, was pretty close after college to going and working in China as an English teacher before I got the chance to work in Ecuador. And I don't know what I would have done if I had gone to China. Probably something cool, but we'll never know. At certain points I thought about maybe going to vet school. Maybe I thought I would be a conservation biologist and work for an NGO. At some point after I got my PhD, I thought it might be really tough to get a professor job, and so I explored careers in data science and biotech. And I didn't go down those paths, and that's fine because I'm really happy about where I am. But I have plenty of friends who did and who ended up in these different places. And so again, the message that I wanted to start out with here is that doing research is a really cool opportunity. And if you're enjoying what you're doing now, I think that's awesome. And you don't have to have a destination in mind. If you do, great. But if you don't, just take it one step at a time. All right. So let's go back to the science part. Like I mentioned, I have a lab here in the Department of Ecology and Evolutionary Biology where I get to work with these really cool people. And we study a few different topics in host-parasite interactions, mostly related to birds and their parasites and pathogens. I'm not going to talk about most of our projects today in the interest of time, but if you'd like to know more about the work that we're doing here locally in the Sonoran Desert, feel free to talk to Marisa. And I'll be talking more about the work that I've been doing for a long time in the Galapagos. And this is couched in the question or the topic of how parasites affect the ecology and evolution of their hosts. We know that parasite and hosts interact in this antagonistic way. The parasites are trying to eat their host. But how does that affect the host? What do they do in response? And to study this, I've been working in an island system for more than a decade. Islands are awesome because they generate unique biodiversity, and there's plenty of organisms that are only found on islands. They're isolated from other continents. They're like mini little laboratories. But the flip side of that is that island species are also particularly vulnerable to extinction. So we know that we've seen a lot of extinctions of biodiversity on Earth, and disproportionately they have occurred in island ecosystems. So for instance, there's about 10,000 birds currently living on this planet, bird species. But it's estimated that humans may have driven more than 2,000 bird species extinct in the Pacific Ocean alone on these islands. So as humans came to these islands, introduced cats and rats and hunted birds for food, that drove what would be right now about a fifth of bird species extinct. So we can have a really large impact. The place that I study birds is in the Galapagos, which is an archipelago off the coast of mainland Ecuador. It's a volcanic archipelago, was never connected to the mainland. Is of course famous because Darwin visited it and his observations helped him form his ideas about evolution. It's also special because humans have been living there for a relatively short amount of time. And so that means that compared to places like Hawaii or the Philippines or Japan, humans have had a much smaller impact on these islands. And that means that we are just in the early stages of understanding how humans disrupt island ecosystems. So most of the archipelago looks something like this. It's beautiful, there's dark volcanic rock and these amazing beaches and a really scrubby, spiny environment to live. But some parts of the archipelago now look like this. And so this is where I stay when I work there. It's a town called Puerto Ayora, and it's got like 20,000 people living there. And the people who live there have over time brought things like cats and rats and other invasive species that are having pretty dramatic impacts on the animals in the Galapagos. So what I will talk to you today is about my PhD work, which I did at the University of Utah, and it was focused on a particular invasive species called the avian vampire fly, or Philornis downsii. So it looks like a fly when it's an adult. It's hard to distinguish from a housefly. But the problem comes, these adult flies, they just eat like fruits and veggies like your house flies would. Anyway, this adult fly lays its eggs in the nests of birds. And these larval stages that you see on the left-hand side, they feed on nestlings, often killing them. It was first recorded in Galapagos nests in the 1990s, so this is a recent introduction. It probably arrived from mainland Ecuador, and it spread really rapidly throughout the archipelago and has become a big problem. And it's a problem because this fly kills more than 50% of the nestlings and nests that it infests. And so here we can see the pathology or the damage that the fly does to the baby birds. On the left-hand side is a healthy baby finch. It's kind of ugly, but it's nice and plump, it's rosy, it's gonna survive just fine. And you can contrast that to the heavily parasitized nestling on the right. That's anemic, it's small for its age, it's got these lesions on its belly and in its nares from where the larvae have been feeding on it, and ultimately this nestling died. And so this is what we're seeing over and over again in the Galapagos. Almost 100% of the nests on the island where I work are infested, and the impact to nestling mortality is quite high. In some cases it's 100%. Well, when I started working in the system, we knew that the fly was bad news, but we also had this kind of asterisk, and that was that observations suggested that one species of bird in the Galapagos, called the Galapagos mockingbird, actually seemed okay with this fly. Like, it didn't really seem to suffer a lot of mortality in the same way that the finches did. And so this is kind of this curious case of a virulent parasite landing in this new environment. The endemic birds of the Galapagos have no experience in their history with this fly, and most of them were dying. But this one wasn't. And so here, I'm going to ask you to talk to your neighbor for a second and speculate about why you think this mockingbird did not suffer mortality due to the fly. What are some reasons that a host might be able to survive when all the other species are really suffering? So 30 seconds of chit-chats. Came up with some hypotheses, and I'd like to show you some of the ideas that we had as researchers. And so if I mention anything that you and your partner discussed, go ahead and raise your hand. Let's see. Okay, so how do hosts defend themselves against parasites? There's lots of potential explanations for this pattern. You know, maybe the mom mockingbirds are picking parasites off their babies. Anyone think of that? Yeah, well, yeah, some people thought of that. Okay, yeah, so that crossed our minds too. What if these baby birds just taste bad? Like there's something about the biology of this particular species that is like the fly is just like, not this one. We don't like the taste of these birds. So somebody in front, maybe that crossed their mind. Anyone else? Yeah, okay. What about the host has some sort of immune response? Yeah, so it could be that there's differences in the biology of these mockingbirds. They have an immune response and some kind of defense. And I think a lot of people here that came to mind because we know that our immune defenses are things that help us fight off parasites, and that's true for birds too. What about, it's just something about the ecology of these mockingbirds, like maybe they choose places to nest where the flies are not common. Anyone? Okay, yeah, so this crosses people's minds, and what you think of as an explanation might depend on kind of your own kind of major or background. And then maybe there's something else that the bird is doing. Maybe it's changing how it lives or how it behaves or how it parents its offspring in response to the parasite. Did that cross anyone's mind? Yeah, some people back there, maybe in front. Is there anything else that you guys thought of that I didn't mention? All right, I'm going to keep moving again for time, but thank you for chit-chatting. So when we think about these different defenses that hosts have against parasites, we can categorize them into 3 different strategies. The first is called avoidance, and this is the strategy of hosts just changing their behavior, where they live, how they act to avoid getting parasitized. So maybe if they're nesting in particular places that parasites are, that would be an example of avoidance. We also have strategies that fall under a big umbrella of resistance, and that includes our immune system, behavioral things to pick off parasites. Those would all be ways that a host can defend itself by reducing its parasite load. And finally, we have mechanisms or a host defense called tolerance, which means that the host is not getting rid of its parasites, but it's finding ways to compensate for the damage done by the parasites. And so we considered all these possibilities and these different strategies to figure out what seems most likely in the case of the mockingbirds. And we sort of decided that it probably wasn't avoidance because they were nesting in the same exact area as the finches. We thought maybe it could be resistance, but no, these mockingbirds actually have a lot of parasites. So when we look at their nests, there's just as many parasites as we find in any other species, so they're not getting rid of them, yet they're still alive. So that pointed us to tolerance. And behavioral observations done by my lab mates suggested that mockingbirds actually increase provisioning to their babies when they're parasitized. And that's maybe how they're able to defend themselves. So by increasing provisioning, the nestlings can recover some of that energy lost to the parasites, and they're able to survive. And so this seemed like a really cool mechanism, increased provisioning, but it also implied a weakness. And the weakness comes from characteristics of the Galapagos. And the characteristic that I want to talk about is the fact that the Galapagos is actually a really tough place to live. Most of the year it looks like this, very dry, scrubby, lots of cacti, but not a lot that one might eat if you're a bird. And the birds have to wait until the rainy season to reproduce. And when the rains come, the landscape really dramatically changes. There's all of a sudden a lot of plants, and that means a lot of bugs that the mockingbirds can use to feed their babies. But both the timing and the duration of the rainy season varies a lot from year to year. And so it means that in dry years, if there's a drought, then the mockingbirds might not have enough food around with which to increase provisioning. And indeed, we knew from the years that we worked there over successive seasons that we had quite a bit of variation in kind of rainy years and dry years. And the years in which my lab mate Sarah found that mockingbirds were tolerant, they actually both happened to be pretty rainy years of normal food abundance. And those were followed by 2 more years that were quite dry. And so that gave us the opportunity to test whether tolerance really depended on food availability and environmental conditions. And so this was kind of a big question that I had in the focus of my PhD research. And so here, I want to present you guys with another kind of like discussion moment and choose your own adventure scenario. So what do you think was the best way to go about this? And what way do we choose to test this question about tolerance? Here's 3 options of potential study designs. They've got strengths and weaknesses. And so chit-chat with your neighbor for another 30 seconds about what you think would be the best way to see if mockingbird tolerance depends on the environment. Talk a little bit about what might be a good strategy in this case. A lot of grad school, a lot of research is designing good experiments, and it's not easy to do because there's a lot of choices to make, and especially if you're working in field conditions, you don't have necessarily control over all the things that you would want to control in a laboratory setting. And so in the field, we have to kind of, take advantage of the options that might be available and try and get at some sort of biological truth in complex situations. So raise your hands if you and your partner thought that option 1 would be a good way to go about this experiment. There's a smattering of people. So the things, we could just go out and study mockingbirds in a natural situation. We could see how many parasites they have, and we would predict that they do better in wet years. How about option 2? Who thinks that one is the right way to go? Literally no one. That's great, I love that. Okay, cool. So option 2 is we're going to do an experiment, manipulate the parasites in the nests of birds, and then we'll just see how things vary between the wet and the dry years. And so that means I think a lot of people like option 3. Raise your hands if so. Cool, okay, so some people didn't vote. Were you dissatisfied with all the options? You just thought no science. I said I'll be in my lab, like this. That's fair, you can always opt out. So option 3 is another manipulative experiment where we supplement the food of nestlings and we see if they do better as a response, and then we know if they died, it's because they really were food limited. So those of you who picked option 3, I think that's a great idea too. In practice, maybe that's what we should have done, but that's also really hard. And so option 3 is a great experimental design, but option 2 is what we actually did in practice, just because that's what we could do at the time. We didn't really know know when we started this experiment that this is what we were going to do. And so in retrospect, some changes could have been made. But let me tell you more about how we did this. So one reason why we decided to treat nests is, and do kind of option 2 instead of option 1, is because we really wanted to compare parasitized to non-parasitized nests in kind of equal footing. And so it's possible that the mockingbirds would have done really well in wet years, like under this scenario. And yeah, of course the reproductive success should be higher if there's more food, but how does that interact with the parasite business? So to get at this question of the interaction between the environment and the parasites, we really wanted to do something to manipulate the number of parasites in the nests. And we can do that by finding mockingbird nests, and then going out there and spraying randomly half of them with an insecticide to kill the phylornis, and the other half with water, which allows them to be infested as a control. And then we kind of measure what's the reproductive success of both groups in each year, and does that depend on whether it's a good year or a bad year. At the same time, we were also taking videos of nests so that we could measure the behavior of the parents. Here you see mockingbird parents bringing food to their babies. And we can measure the nestlings so that we see how they grow, whether the mass differs between infested and uninfested nests. And then ultimately we assessed whether they survived, so whether they successfully fledged from the nest. And then after the mockingbird nestlings left their nest, we can take down the nest and count how many parasites are in them. So we know exactly whether our treatment worked, and more importantly, how many phylornis parasites were actually in each nest. And so what we found was that in each year, parasitism definitely has a toll on the nestling. So on your y-axis here is hemoglobin concentration, which is a measure of anemia. And we're looking here at permethrin, which means not parasitized nests in pink, and comparing them to those nests in blue that were parasitized. And in each year, we see a significant difference in hemoglobin concentration. So these larvae are sucking the blood of these nestlings, and it shows. However, the ability of nestlings to compensate varies among years. And so now we're looking at the mass of nestlings. Mass is on the y-axis, and each year that we did this study is on the x. And we had 2 rainy years, which were the first 2, and that was followed by 2 dry years. And again, we have colors for nests that were parasitized in red, or sorry, not parasitized in red, and parasitized in blue. And so in the first 2 years, even those parasitized nestlings, even though they were losing blood and they were kind of anemic, they were still growing at pretty similar rates. However, in the subsequent 2 years that were dry, we saw a pretty significant difference in size. And so that meant that they were losing blood to the larvae, they were not recovering it, they were not keeping up that growth trajectory that they should have been for little birds who were trying to get out of the nest. And ultimately, this translated into real differences in fledging success. So this on the y-axis is the percent of nestlings that fledged out of each nest, and each bar represents an average for the treatment for the year. And we had 2 years, again, that were rainy, 2 years that were dry, and the bars, the color of the bars shows our treatments. So in the first 2 years of the study, there was no significant difference in fledging success between treatments. These mockingbirds were completely tolerant to the parasite. Even mockingbirds that had 100 parasites in their nest, they were still surviving at equal rates as those nests that didn't have any parasites. But in the dry years, these mockingbirds lost their tolerance. And so under dry conditions, when these mockingbirds are not parasitized, they still do pretty well. So those pink bars are pretty similar across years. So even when food is kind of limited, under normal situations, the mockingbirds are adapted to this environment. They can still figure out how to fledge their babies. But when they're parasitized by this new, virulent, really challenging threat, they can't deal with both the drought and the parasites. And as a result, we see this difference where the mockingbirds kind of lose their tolerance, and we see a real difference in fledging success between parasitized and non-parasitized birds. And when we looked at our videos, we also found that the provisioning rates kind of corroborated this story. And so when we measured the amount of time that parents spent feeding their nestlings each year, we saw that the more time that parents overall spent provisioning their nestlings, more tolerant they were. And this kind of pointed us again to the environment and thinking that the amount of food that mockingbirds have access to in each year really matters. So putting it all together, we see a story where environmental conditions mediate tolerance of mockingbirds when they're dealing with this new invasive parasite. It's when there's a lot of rain, that means that more vegetation grows, there's more arthropods for the mockingbirds to eat, and they're better able to compensate for the damage done by this parasite. But when things get a little bit stressful, if they don't have any parasites to deal with, the mockingbirds are still fine. But if they have to deal with the added burden of this invasive parasite, then they really suffer some big fitness losses. And so the kind of recap of this vignette is that this project was all about how parasites affect the ecology of their hosts and how a novel introduced parasite might, what we can learn about how animals deal with the arrival of a novel introduced parasite. And the good news is that naive hosts are not always defenseless. So these mockingbirds have never seen this parasite before. They don't have a specific antibody or particular defense that helps them deal with